Hello everyone, and welcome to today's webinar on bioanalysis of small and large biological molecules, improving selectivity, sensitivity, and reproducibility. My name is Roland Meesters, and I will be your host for today's webinar. I'm the editor-in-chief of the Journal of Applied Bioanalysis, and I'm excited to be hosting this session for you today. I'm pleased to introduce today's speaker, Dr. Kwasi Antwi. Dr. Antwi received his PhD in 2006 from the Department of Chemistry at the University UMass Amherst. In 2009, he completed his postdoctoral fellowship at the Arizona University. In 2013, he joined Thermo Fisher Scientific as application specialist for a mass spectrometric immunoassay group. At his current position at Thermo Fisher Scientific, Dr. Antwi has developed several immunoassays and LCMS methods to study biomolecules, including monoclonal antibodies, antibody-drug conjugates, fusion proteins, insulin, and many, many more. His work has been published in application notes, which are being utilized by the biopharma industry to study those molecules. Before I hand over the mic to Dr. Antwi, I have few housekeeping items to cover about this webinar. First, today's webinar will be later available on social media platforms. We will inform you about this possibility also later. Next, we love to hear from you during the webinar. So, if you have a question for our speaker, please feel free to use the chat tab. This you find on the right side of your screen. We'll be answering some questions at the end of the webinar. So without any further ado, I'd like to kick off this webinar by welcoming Dr. Kwasi Antwi.
Thank you so much, Roland, for such a beautiful introduction, and also thank all of you for coming on the line today. Today, I'm going to speak about bioanalysis of small and large biological molecules, improving selectivity, sensitivity, and reproducibility. So in this presentation, I'll be talking about the biological molecules that we study in our labs and in other several labs. What are the traditional methods that are used to study these biological molecules? And I'll also go into the hybrid HPLC mass spec methods that we use here in our lab and other labs. We call that the mass spectrometric immunoassay, which we pronounce as MESA. And I'll also walk you through some of the things that you can do to improve your bioanalysis data, such as sample prep, immunocapture, ligand choice, and also the LCMS method that you can also develop to improve your signal. And then I'll walk you through some conclusions. So here's a list of most common biological molecules that we study in our labs and also in various labs out there. And when we look at these biological molecules, the most common techniques that are usually used to study them are the ligand binding assays. And as you can see here in the presentation, when you have your ligand binding assay, you're always going to have your ligand, which is coupled to a surface, and then use that ligand to capture an analyte, and then come in with the horseradish peroxidase molecule to detect the complex, and then the signal that is produced, you detect that which is correlated to the quantity of the analyte that you have in your sample. And here I want to also mention that this is indirect detection of your molecule. The beauty of this ligand binding assay is that it's simple to perform. It's so easy compared to other methods out there. It gives you the quantitative information that you want, and it can be very sensitive. But the challenge that you can also encounter in this LBAs is the fact that because everything is indirect detection of your molecule, any non-specific binding in the background can also interfere with your analyte signal. Another issue you also have with the LBAs is the fact that they lack characterization information. What I mean by this is that you cannot sequence your protein using the LBAs. You cannot extract any modification information such as the post-translational modification in your molecule. You're also going to encounter matrix interference, which is going to affect your selectivity. And usually the calibration curve that you generate in LBAs are non-linear, so that can also affect the quantitative information that you can extract. And also the ligand binding assays have drug interference issues, so that also affects the specificity of the ligand binding assays. So that also leads us to, what other methods out there can be utilized to address some of these challenges that I just discussed. So I would like to walk you through, hybrid LCMS methods available out there. Today, I'm going to focus more on the mass spectrometric immunoassay. As I already mentioned, we pronounce this as MESA, and I'll walk you through the workflow of MESA. What is MESA? What does it do? What are the things that you take into account when you're doing MESA? First, it's a hybrid LCMS, so that means you need a ligand that will go on the surface, and then you're going to use the ligand to then capture your analyte and then elute your analyte and analyze it on the LCMS. This technique is very powerful because it gives you qualitative information such as the characterization information, where you can sequence your protein and also extract post-translational modification and other modifications that your protein or your analyte can undergo. There are some individuals or labs that are also interested in looking at degradation products, such as doing catabolism or metabolism studies. So MESA can help you do that kind of study. And it is also highly specific in the sense that, unlike the LBAs, for MESA you are doing direct detection of your analyte, so you know what you are detecting, and it gives you the quantitative information, where in MESA you are also able to extract quantitative information and draw a calibration curve that is more linear than what you obtain in the LBAs. It is a very sensitive method. You can go as low as even 0.1 femtomole detection for some of the analytes, and it doesn't take too long to be able to develop your method. And you have the capability to be able to multiplex your analytes. In other words, you can detect more than one analyte in your sample, and it is less susceptible to drug interference, in the sense that your LCMS can help with the separation of molecules. So how does the workflow work in MESA? So as you can see over here, you have a micro column that is housed in a tip. This is a pipette tip. And on the surface of this is a porous micro column. So on the surface of the micro column, you can put some other chemistry such as streptavidin. You can put protein A, protein G, protein AG. You can also put your own affinity ligand on the surface. So In this example, I have streptavidin, which is coupled to the surface, and then use that streptavidin to bind biotinylated capture select. Capture select is an anti-IgG Fc binder. So using the tip here, I can now go into my sample and then capture therapeutic monoclonal antibody. Once I capture my analyte, I can then go ahead and elute and then detect the intact analyte on the LCMS. I can also digest the analyte and then detect the peptides that come off in the digestion protocol. So when it comes to the tools that you need to do MESA, we're looking at down here the pre-analytical tools. You can do the pre-analytical part on the Tecan Freedom Evo or the Thermoscientific Verset. These are instruments that will let you do automation of the pre-analytical steps, and also you can analyze 96 samples at a time. If you want to do a few samples, in most cases during the method development, you don't have to run a whole lot of samples. You can also use the Novasi multi-channel pipette, which will let you do a few samples, up to 12 samples. For the analytical detection, we use the Vanquish UHPLC system, and also couple that to the Q-Exactive mass spectrometer to do the detection, and then use the Biopharma Finder software to do the data analysis. It can be quantitative or qualitative data analysis. So MESA can be utilized in various research labs. So here we have the biopharmers, we have the clinical research labs, we have the sports anti-doping labs. Those are areas that use biological samples. So you can use the MISA workflow to obtain reliable and accurate and also reproducible data for your bioanalysis. So what are some of the things that you can do to improve your data when you're doing, hybrid LCMS? And for this purpose, I'm going to focus more on the MESA workflow. So the first thing I want to talk about here will be the effect of sample prep techniques. So I want to focus on these two techniques I have here on the slide. These are two common sample prep techniques that are utilized in bioanalysis. The first technique is the pellet digestion solid phase extraction. What this sample prep technique does is that you make a pellet of your analyte, and then you take the pellet, redissolve it, digest whatever comes out in the redissolution, and then go ahead and capture your analyte on the solid phase column, and then run the eluate on the LCMS. On the second technique, which is the immunocapture technique, here you have two ways you can do the immunocapture. You have the tip-based, such as the MESA technique that I just described, or you can also do the beads-based, where you put your immunoaffinity ligand on beads and then use that for the capture of your analyte. So we have a study that was done by Oturas Analytics. So what they tried to do here was to analyze ADC, or develop workflow for ADC in plasma. And the first method that they chose was the pellet digestion SPE method. And when they developed the method, the lowest concentration they could detect was the 100 nanogram per ml. And their goal was to be able to detect 10 nanogram per ml. So they didn't achieve the goal that they set, using the pellet digestion and the SPE analysis. So then they moved on to test the MESA workflow. And using the MESA, they were able to detect as low as 10 nanogram per ml in the sample. This is a tenfold increase or improvement in the signal. And you can also see in the two chromatograms that the MESA chromatogram looks, much cleaner, less background compared to the SPE method. This data was presented by Shane Needham this year, January, in Bioanalysis webinar series. The second study here was also done by Covance. So what Covance was interested in doing here was to be able to use bioanalysis workflow to detect insulin in serum or in plasma. So first they tested the SPE. The target was to be able to hit about 70 picogram per ml of insulin in serum or plasma sample. When they used the SPE, the lowest they could detect for glycogen was 200 picogram per ml. So that was not enough to, get the job done. You can also see all the background here in the chromatogram. So that wasn't pretty at all. So then they switched to the MESA workflow. And in the MESA workflow, they were able to detect as low as 50 picogram per ml, which met the expectation that they set for this study. So you can also see from the MESA that the background in the LCMS was so clean, and that helped to improve the signal. Then in the study here, this was done in our lab. So we've already compared the SPE to the MESA workflow. How about comparing two immunocapture workflows? So here we're comparing MESA to the BEATS workflow. And what we observed was that when we use MESA, we are able to reduce the background in the LCMS by 50 times compared to the when we use the beats. So you can see here that the beats had huge background. As a result, that killed the signal. So when we went to the detection of the analyte of interest, we were able to improve the signal for the analyte using the MESA workflow compared to what we detected in the BEATS methodology. So this again is showing that the MESA workflow can minimize the background of your LCMS and improve your signal. The other information that we also observed in this study was the fact that the error bar on our MESA results, was much better than what we observed with the BEATS method, meaning that the MESA workflow was more reproducible than what we observed using BEATS. And I want to also highlight here that this data is not only been observed in our lab, there are other labs out there that have also observed, similar nature. So MESA gives you better detection than what you observe for your BEATS methodology. And also because of the fact that we see better, background in the LCMS, we also realized that it helped us to extend the MESA helped us to extend the lifespan of our LCMS columns, and we didn't have to, clean up the LCMS more often as we did with other methods. What's the next thing that you look for and improve to be able to achieve better data for your bioanalysis. The next thing I want to talk about is the effect of the sample matrix. The sample matrix can impact your data. So this is a study that came about when a customer contacted our lab and wanted to do this analysis where they want to put the biotinylated TNF-alpha on the tip surface here and then use that to capture the adalimumab and infliximab as the analytes. The next question that they asked when we went through the discussion was, do we use plasma? Do we use serum? For some of you who may be new to this, when it comes to plasma, you are using anticoagulants to obtain your plasma. When it comes to the serum, you don't use those anticoagulants. So there's that difference right there. So which one gives you better results? So we needed to look into that. But let me also mention that the analytes that we captured here went through trypsinization. So we did the trypsinization to create our peptides and analyze the samples on the thermoscientific Vanquish and the Q-exactive mass spectrometer. The column that we used here was the ProSwift RP4HLC column, the 1x250 column. So when we finished the discussion, we decided, all right, let's go ahead and look at the matrix effect on the two analyzed, the infliximab and the adalimumab, and see any matrix effect on these analytes. So we first looked at the blank plasma versus blank serum to find out, is there any matrix effect in analyzing the infliximab. And as you can see here in the data, look, these are the two peptides that we monitored for infliximab detection. And we didn't detect any background, whether using EDTA plasma or the sodium heparin plasma or the serum. So that means when you want to analyze infliximab, it doesn't matter which matrix you use, you're still going to get good data. Then when we went into the adalimumab study, these are the two peptides that we monitored for adalimumab detection. And we realized that even in the blank plasma and the blank serum, we detected a signal in EDTA plasma for both peptides that we monitored for adalimumab. So that tells you that if you're going to detect adalimumab, EDTA, it's not going to be the choice of matrix that you want to use. The choice of matrix would be the sodium heparin, which in both peptide detection, we had great results, low background compared to using EDTA or the serum. So that tells you when you want to develop your bioanalysis, don't just jump on, I'm going to use serum or I'm going to use plasma. You want to be able to, do some investigations and see which one will work best for you. The next thing that you can also do to improve your bioanalysis data, and for that matter, the sensitivity, is the affinity capture ligand. Most cases, what we do is that we just go in and say, okay, I'm gonna use protein AG for my analysis. Why do you want to use that? Because that's what I have available. But sometimes you never know what you are missing for not using other ligands. So we tested out some ligands, as I have listed here in the first column of the table. And the difference between these ligands is the fact that they differ in molecular weights. So as you see in the table, the molecular weight difference goes from 13 kilodalton to 150 kilodalton. What is common about them is that they are all FC binders, so they all bind at the FC region of the antibody. The sample that we use for testing the capture characteristics of these ligands was the ADC spiked into rodent plasma. And again, we use the thermoscientific Vanquish LC system coupled to the Q-exactive mass spectrometer for this analysis. And here what we did, this is the protocol that was followed. So everything was processed, the pre-analytical steps were all done using the thermoscientific Versed or the TCAN Freedom Evo. And as you can see, the steps are here. We loaded the ligands, the individual ligands on the tips, and then loaded the tips on the automated liquid handler, and then went ahead to do the pre-wash of the tips, then captured the analyte of interest, in this case the ADC, wash with PBS to remove any non-specific binding, and then wash with water to remove any salts, and did the elution in acidic solution, and then the eluate was analyzed on the LCMS. This is the typical example of data that we observed. So over here, you have your total ion chromatogram. And when you extract this region of the chromatogram, what you see here is your mass spectrum, which represents the ADC. And when you deconvolve your spectrum here, the mass spectrum, you obtain the second spectrum, which we call the deconvolved spectrum, which shows you the analyte without any drug attached. That's the antibody without any drug attached. And when you have one drug attached versus two drugs versus three, and so on and so forth. This ADC was created using lysine chemistry, so that's why you see several multiple drugs attached. And in this data, you see the beauty of the Q-exactive mass spec. So this mass spec is able to analyze the ADC intact. You don't have to do any digestion. The mass spec is able to acquire the data. It gives you the wide M/Z range, and it's also high resolution mass spec. So you are able to see isotopic peaks and stuff like that, to be able to gather a lot of information. So Q-exactive, we love it for this kind of analysis. When you sum up all the peaks that you observe here in the deconvolved spectrum, you are then able to do further analysis, as I'm going to show you in the next slide here. So here we sum up all the individual peaks and then plotted the sum of the peaks, the peak intensities, for each of the ligands that we analyzed or we studied. Before I show you the information we gathered from the data, I want to first talk about the neat sample peak here that you see. So when it comes to the neat sample, this is not sample that was analyzed through MESA. So we took just the pure ADC, prepared the same concentration as was used for the capture, and then just ran it straight on the LCMS to see what signal that we will obtain. And here, relative to this neat sample, we can then figure out the capture characteristics of all the other ligands that we studied in this particular work. And here we can see that relative to the neat sample, the capture select was able to extract about 50% of the ADC, followed by the protein AG that was able to capture about 40% of the ADC. The anti-idiotype was able to capture about 30%, and then the mouse IgG was able to capture about 20%. What you see here is that as you go from left to the right on the plot, the molecular weight of the ligands are increasing. So we went from 13 kilodalton all the way up to 150 kilodalton. What is going on here is that we believe the smaller molecular weight ligands are able to have better accessibility to the FC binding region and therefore able to bind the ADC better than the larger molecular weight ligands. So we think maybe it's some steric hindrance going on between the large ligands versus the ADC binding. So that's why you losing binding affinity. So overall, we saw better binding, better capture with the low molecular weight ligands versus the larger ones. When it comes to the sensitivity, the plot here doesn't look all that great. This is not how it looks like on the original slide, so pardon me for that. So when it comes to sensitivity, we are able to detect lower concentration of the ADC using the capture select compared to all the other ligands that we studied here. Also, when it comes to the dynamic range, we are able to see wider dynamic range using the anti-idiotype compared to all the other ligands that we studied here. So all this data is showing us that don't just jump on one ligand and use it all the time. You want to always test out other ligands out there and always choose the one that works better for you. In this particular study, if our goal was to hit lower sensitivity or better sensitivity, our choice would be the capture select. If the goal was to cover wider range, then our choice would be the anti-idiotype. The next thing you can also do to improve your data is going to be the LCMS method. So when it comes to the LCMS method, I'm going to show you what methods are available on the Q-Exactive instrument, because that's what we have in our lab here. And it works great. We love it. So these are three methods I'm going to talk about, but there are other methods that are also available on the Q-Exactive mass specs. So the first mode that you can use for your LCMS method is the full MS mode. What does that one do? When it comes to the full MS mode, you are able to scan pretty much all the ions that your the mass spec will enable you to detect. That's you are only limited by the M/Z range on your instrument. So if your instrument, especially on the one we use here, you can go as high as 4,000 M/Z. So you can capture all ions from 50 to 4,000 M/Z. So that's great. It helps you to detect your analyte. It helps you to also see any modifications that may occur in your analyte. So that's great. But the downside to this method is that because you cover a wide range of M/Z, your instrument doesn't scan really fast, and therefore you're going to suffer in sensitivity when you use this method. The second method you can use on the Q-Exactive is the SIM method. So the SIM method is similar to the full MS method. The only difference here is that you narrow the M/Z range that you want to detect. So therefore, your instrument is able to scan much faster and become more sensitive. But again, the downside to this method is that you're going to limit your ions to the range that you select. That means all ions that fall outside of the range that you select on the instrument will not be detected. Then the third method that you can also use for your LCMS method development is a PRM. So PRM is where you have pre-selected precursor that you just want to focus on the instrument, for detection. So you line up all these precursors, and if you don't put, any precursor on your list, that precursor will not be detected. So you are going to have better sensitivity with a PRM because you limit the instrument to only scan a few ions. But that also means that if you came back and said, oh, I just realized my analyte has a modification, and you didn't put that modification in your list, your inclusive list, then that's it. You cannot go back and detect that ion. So that's the downside to this method, but it's a very sensitive method, and you also do MSMS mode with this method. So you fragment, you save all the ions, unlike say the triple quad where you fragment and only detect one or two or three daughter ions. In the PRM, you're able to store all the ions, the daughter ions that are created, and then you come back later and then select which daughter ion you want to use for data analysis. For example, if you want to do quantitation. So we have one of our colleagues in Thermo Fisher, Kelly Murphy, who wanted to analyze insulin. So when it comes to insulin analysis, you're going to have, differences in the molecule, especially the molecular weight. The differences, in the insulin analogs is mainly due to substitution of amino acids. But when it comes to the humalog, which is also called the lispro, the difference between that and your human insulin is that two amino acids here in black are flipped. So that means the molecular weight for humalog is still the same as that of human insulin. So it makes it challenging to detect the humalog in the presence of human insulin, the endogenous human insulin. When it comes to the levemir, you are adding a hydrophobic group called the myristic acid to that. So it makes that insulin analog very hydrophobic. So here's the protocol that Keeley used to do the insulin analysis. So here he's using the thermoscientific Versed to do all the pre-analytical steps. So as I have already shown you in the ADC analysis, first you couple the anti-insulin antibody to the tip and then load the tips on the Versed and then pre-wash the tips with PBS and then go into your sample to capture the analyte, in this case the insulin analogs, then wash off any non-specific molecules and then wash all the salts with water and then elute into acidic solution, then run the eluate on the LCMS. So in Keely's work, his goal was to be able to detect 25 picogram per ml of the insulin. And when he used the full scan, he was not able to achieve that goal. So the best sensitivity he was able to achieve was 90 picogram per ml. When it comes to the upper end of the curve or the range that you see here, I'm not going to spend much time to talk about it, because you can always push this upper end up. So you are not limited by the 5,720 that you see here. But the lower end of the curve is what is important in this presentation, because if you are using full scan and your goal is to detect 25 picogram, the full scan will not be able to help you to achieve that. But when he switched to the SIM, for the same two insulin analogs, he was able to achieve the 25 picogram per ml. So that was great to see that different LCMS method can help him achieve the goal that he set for his study. When it comes to the determer, the detection here is much higher than what he observed for the other insulin analogs, because the determer is so hydrophobic, and therefore it reduces the ionization efficiency on the LCMS. Then he also tested out the PRM method. So with the PRM method, he pretty much observed the same data as he observed for the SIM. The additional information that he was able to extract from the PRM was the analysis of the insulin lispro, or the humalog. So if you look at the two methods that I already discussed, the full scan versus the SIM, he was not able to use those two methods to distinguish the humalog or the lispro from the endogenous human insulin, because they have the same mass. But when it comes to the PRM, where you fragment your insulin, he was able to focus on the daughter ions that were created in the fragmentation to distinguish the lispro or the humalog from the endogenous human insulin. And that was very helpful, knowing that this method was able to help him do that study. So when it comes to the LCMS method, you always want to set your goal and ask yourself, what do I want to achieve? And then use the method that will help you to achieve that goal. In this case, if the goal was to achieve separation between lispro and humalog, the PRM was going to do the job. If it was just to detect and not do any distinction between lispro and endogenous human insulin, then SIM will get the job done. So in conclusion, I would like to say that the sample prep techniques can hurt or help your method, for analysis of biomolecules. So you always want to investigate the sample prep technique that you choose for your method. And also, when it comes to MESA, I just showed you here that you can use MESA to improve the sensitivity of your bioanalysis. Then when it comes to the immunocapture ligands, if it's possible, you have the money and the time to investigate, I would strongly recommend that you investigate more than one ligand and see which one works, best for your study. And then the LCMS method, you also want to investigate which one fits, the goal of your study. So at this point, I would like to thank the following people: Chad Christensen from Oturas Analytics. So Chad was the one that did the work where he compared ADC capture on SPE versus MESA. I also want to thank Michael Blackburn. He was the one that did the study that compared insulin on SPE and MISA workflow. And Keely Murphy was the one that did the insulin work on different LCMS methods. And I also want to thank my colleagues in MISA, Chris Ross and Eric Niederkoffler. If you want to learn more about MISA, you can visit the links here on the left. And if you have any more questions, if you want to address certain specific issues that were not discussed here, you can also send email to the email address on the right here. And on this note, I would like to say a big thank you to all of you.
Thank you very much, Dr. Andre, for this very interesting presentation. I think we have, let's say, some 5 minutes left for some questions. I don't know, are there any questions from the people in the audience? If you have a question, please use the chat tab that you find on the right in the blue ribbon. I see for the moment there is no question. Maybe I can answer, maybe I can ask you something.
Yeah.
That's maybe also interesting for the audience. I'm wondering, is it difficult to validate a bioanalytical method using MESA? Is it completely different than, let's say, validating a general analytical method?
I would say no, because we have, for instance, FDA guidelines came out 2008 version, 2018 version, and it outlines, all the steps that you need to follow in order to validate your, biological analysis or, bioanalysis, if I may call it that. So following those, guidelines, there's nothing in there that, MESA will not be able to help you achieve. So it's not going to be difficult to use MESA for bioanalysis validation.
Okay, thank you. I see there's a question. A question came in.
Yeah. So I have someone asking, has MESA been tested to diagnose antibodies against specific microbial pathogen? Not sure what the person is asking about here. Are you asking about whether it's using antibody on the tip, you are able to capture microbes? So if that's the question you're trying to ask here, we had one customer who was trying to do this kind of study where they're going to put the antibody on the surface of the microcolumn and then use that to capture the microbial or the microorganisms. And it turned out that the pore size that we have in the microcolumn was smaller than the size of the microbes. So just for, because of that issue or challenge, the customer was not able to use MESA, but it wasn't because they were not able to capture the microbes with the antibody. It was just the pore size in the microcolumn that didn't allow them to flow the analyte through the microcolumn.
Okay. I see there's no second question. Okay, then I think we're done. So
okay.
I'd like to thank you again for the very nice presentation, very informative.
Okay.
And I also like to thank the audience that visited us today during this webinar. And I will see you next time. Thank you very much.
All right. Thank you.
Bye-bye.
Bye.